Okay, so shall we start with Christine Jones? I don't think Christine needs any introduction. It is her third year speaking to BioFarm, and I know that she has consulted with many farmers here in Ireland as well. But we're going to pass over now to Christine. Hello, Christine.
Hello, Ella, and hello, everybody.
Christine, where in the world are you coming to us from?
From Australia.
Fantastic. What time is it there?
Just after midnight.
Right. Well, thank you very much, Christine, for taking the time to join us again. Your words are hugely listened to by the BioFarm audience. So I'm going to pass it over to you now, and then afterwards, hopefully, we'll have time for questions.
Thank you very much, Ella. I'm conscious of the clock, and I will leave plenty of time for questions today. So I'm just going to get straight into my presentation, I think. Can we see that? The soil sociobiome?
Thank you. Looks great.
Yes, can you see that?
Yes.
Great, good. Let's go. So on Wednesday, I spoke about what's in a seed and why does it matter. And today is part 2 of the soil sociobiome, which is about the sequestration and stabilisation of soil carbon. I do apologise that this is very academic, but I think one of the challenges that we have in agriculture is that when a farmer obtains some amazing result, the academics turn around and often say, well, where's the science? So I guess my role in this is to bring forth some of that science and show that there actually is a lot of science behind what we're seeing in the field. So the first thing about soil organic carbon, which is often just denoted as SOC, is that it is a combination of carbon and various other things. So it's not just like 100% carbon. It's about 60% carbon and 6 to 8% nitrogen, and it's somewhere between 32 and 34% of that soil organic carbon molecule is soil minerals. So soil organic carbon is something that is formed in situ in soil. It's a biological process, and we have to be very conscious of the biology and particularly the microbes that are involved in that process. We also have to be very aware of the fact that if we're just looking at this from a chemistry point of view, is that the carbon and the nitrogen are bound together in the same molecule. And this tends to be overlooked for some reason. We talk about carbon a lot and we talk about nitrogen a lot, but we forget that in nature that carbon and nitrogen are actually bound together in the same molecule, organic carbon and organic nitrogen. So when the levels of soil organic carbon increase, the levels of soil organic nitrogen also naturally increase, and vice versa. So when levels of soil organic carbon fall, levels of soil organic nitrogen fall. And those falling levels of organic nitrogen are what prompt farmers to apply inorganic nitrogen fertiliser, which is a different thing to organic nitrogen. And this has numerous adverse effects. I'm just going to briefly mention a couple of these. One is that inorganic nitrogen fertilisers, so I'm talking about things like urea and ammonium nitrate that contain nitrates and ammonium, obviously. Nitrate is highly mobile in soil. It's very rapidly lost either through leaching or volatilisation. Only 10 to 40% of the nitrogen that's applied to plants is actually taken up. So 60 to 90% goes somewhere else, causing immense environmental damage. And the other thing is that because it's so highly mobile, it's very rapidly transformed and it doesn't accumulate in soils. Farmers apply nitrogen, they lose most of it. It causes damage somewhere else, causes a huge amount of damage to their bank account because it's so inefficient. And then there is no bank of nitrogen, so you have to apply it again. And a recent EPA report, which was from July this year, showed that nitrate levels were unacceptably high in 47% of Irish rivers, 25% of Irish groundwaters, and 20% of Irish estuarine and coastal waters. And if we're talking about climate change and greenhouse gas emissions, when we look at the inorganic nitrogen fertiliser supply chain, this is like everything from the production and the transport and the application of inorganic nitrogen fertiliser, has been shown to emit around 1,250 million tonnes of CO2 equivalent. This was in 2018. This is the most recent data that we have. And just for comparison, so just remember that number, 1,250 million tonnes of CO2, the global commercial aviation industry emitted 900 million tonnes in 2018 of CO2 equivalents, far less. We think of aviation as something that's a huge polluter in terms of greenhouse emissions, and yet the supply chain for nitrogen fertiliser emits far more greenhouse gases than commercial aviation. So the best way to reduce our carbon footprint would be to stop using nitrogen fertiliser. Well, why do we use it? Why is it that we're using this industrially produced compound that has enormous adverse environmental effects, including being a significant contributor to greenhouse gas emissions? Because when we actually look at the data, and there's been quite a lot of discussion about dairy and the dairy industry over the last few days of this conference, there is a very poor relationship, if any, between the amount of nitrogen fertiliser that is applied and the amount of milk produced. Now, this is Australian data. The red line along the bottom here is the amount of nitrogen fertiliser. The red line along the bottom is the amount of milk produced over time. And this black line is actually the amount of nitrogen fertiliser that's being used. Now, if you can see a relationship between those two lines, you're a better statistician than I am. This very slight increase in milk production that we've seen over time has actually been due to improved genetics of the dairy herd and also just improved efficiencies in milk production in general. No relationship whatsoever to nitrogen use. Increasing the amount of nitrogen that's applied will actually increase the biomass of the forage that's produced, but it does not contain it doesn't have any nutritive value for cows. So although a rising plate meter will show you that there's more biomass there, it doesn't relate to milk production. So the use of high analysis nitrogen also reduces soil carbon. And we see that there is an inverse relationship between soil levels of inorganic nitrogen. So I'm talking again about things like nitrates and nitrites and ammonium and root exudation. So having a lot of inorganic nitrogen floating around in the soil inhibits exudation from plant roots. And the classic example of that is, this is a wheat plant. You can see the seeds are here. It has had 80 units of N placed under the seed when it was drilled in at the time of planting, and all of the roots that are coming out of that seed, the seminal roots, are totally bare. They have virtually no exudates coming out of them, and so they haven't formed riser sheaths. This farmer put compost on the surface of their soil, and you can see that the roots that are coming out of the crown of those wheat plants have beautiful riser sheaths. They are producing lots of exudates, and they are forming a relationship with soil microbes. So this is just, this is not an over the fence comparison or anything. This is just like in the same time and place, we can see the incredible inhibitory effect of nitrogen on root exudation. And here's a close-up photo of a plant root. I think I did show this one last year as well, with droplets of exudates coming out. You can also see that there are fungal hyphae, just like cradling those droplets of exudates. These exudates are the beginning of stable soil carbon sequestration, which is what I'm going to talk about next. So root exudates are actually required to build soil carbon. And when we don't have those exudates, we lose soil carbon, and then that results in a greater requirement for inorganic nitrogen. So in terms of nitrogen fertiliser, the more we use it, the more we lose it. The farmers are on this incredible treadmill of applying nitrogen, losing soil function, losing soil carbon. Most of the nitrogen's going somewhere else and causing a problem. massive greenhouse gas emissions from, as I said, from the production and the manufacture and the transport and the application of nitrogen. And then soil deteriorates, soil function declines, and then farmers are in the situation of having to use more. I can't remember what the number is off the top of my head, but I think something like soil nitrogen fertiliser use has increased 800% or something in the last 20 years. So how can we rapidly build soil carbon so that the requirement for inorganic nitrogen no longer exists? Obviously, if we can turn that around and find ways to build soil carbon, there won't even be a requirement for inorganic nitrogen. It's not a matter of using precision agriculture and all these kinds of things of like a more efficient use of nitrogen or coating the nitrogen so that it doesn't, that it's not so water soluble, and all these incredibly complex arrangements we come to try and make nitrogen less of an issue. We actually don't need to use any nitrogen at all. So we need to change the way we farm so that the requirement for inorganic nitrogen simply no longer exists. So let's talk about soil carbon. The research into soil carbon has traditionally focused on the quality and the quantity of plant biomass inputs. There are literally thousands and thousands of papers on soil carbon that talk about how many tonnes of organic matter you have to add to soil to increase soil carbon by so many percent. And it's actually all totally irrelevant because that is not how soil carbon is formed. So recent research has shown that stable soil carbon is actually dominated by dead microbial products and residues, and this is called microbial necromass. In other words, even though microbial biomass, like the living biomass of microbes, probably contributes around about 5% to total soil carbon, when microbes die, what happens to their bodies is incredibly important, and they turn over very, very quickly. So if there's a lot of microbial biomass and they're living and dying, living and dying and living and dying, what happens to all the dead microbes? Well, under appropriate conditions, those dead microbial bodies actually become microbial necromass, and they're entombed in the soil as stable soil carbon. The first person to really talk about this in any great detail was Liang back in 2017, so 4 years ago now. He talked about root exudates and how they actually stimulate microbes in the soil. And then he talked about this entombing effect that he called the microbial carbon pump. And the end result of this was microbial necromass. But at that point, back in 2017, no one was really quite sure what was going on there. But we did know that something was happening to turn very labile carbon that comes out of roots in the form of sugars and other carbon compounds, which you would think that microbes are just going to utilise that and it would be gone in a very short amount of time. But actually, we hadn't considered, well, what happens to the microbes when they die? And they're entombed and they become microbial necromass, which is what we used to call humus. This is just another diagram of this, the same concept. We have a plant, we have the root inputs going into, we've got this living biomass of microbes that we're fairly well aware of because you can see those under a microscope, but then they become dead microbes and become entombed in the soil, and they're going to be occluded with minerals, and that's when they become stable. They can become stable for thousands of years once this entombing effect takes place. And as I said at the beginning, stable soil carbon is around 60% carbon, which is the carbon that came through this pathway from photosynthesis and microbial activity and microbial necromass, and about 6 to 8% nitrogen, and then the other 30% is minerals. It's actually a part of the stable carbon molecule. And it's very, very important that in this entire process that the nitrogen that also becomes part of the stable carbon molecule is fixed biologically. If we add nitrogen to the system as inorganic nitrogen, so if we add it as urea or nitrogen, what's that one you use? Forgotten the name of it now, ammonium nitrate, which we don't use in Australia, but I think you use in Ireland. then the plant will not be exuding sugars to feed nitrogen-fixing microbes, and so this whole process basically comes to a halt. This is just another diagram showing the same thing. We have our plant inputs coming into microbial biomass. It's just showing it in another way. When they die, they will become necromass, and then depending on what actually happens after that, what kind of minerals are in the soil, things like iron and aluminium are very important for the stabilisation of microbial necromass into this mineral stabilised necromass. And once we have this in the soil, we have a very, very stable form of soil carbon. So the fungal and bacterial necromass obviously is going to increase with the living microbial abundance and diversity. So the main thing that we need to concentrate on in terms of building soil carbon is having greater microbial abundance and greater microbial diversity, because those things are going to feed back into necromass. And results from the Yena biodiversity experiment, which I have mentioned before, showed that root exudates increased, microbial abundance increased, and microbial diversity increased with plant diversity. You'll all be well and truly sick of hearing about plant diversity by the time this conference is over. There was a question I was asked on Wednesday, which I must apologise to Janine if she's listening, because she said, what about the 16 plant species? I didn't actually mention them on Wednesday, but one of my presentations was replayed on Monday, in which it would have been mentioned about this experiment, because in the Yena experiment, they looked at 1, 2, 4, 8 or 16 plant species. But what is really important about that was that they were from 4 functional groups. So if you had 4 species, there would be 1 species each from the grass group, from the legume group, from the tall herbs group, and from the short herbs group. So tall herbs could be something like chicory. At the moment here in Australia, we have chicory growing up over our heads. It's like just gone crazy this year. And short herbs would be things like plantain. So 4 is 1 from each of 4 functional groups, 8 is 2 from each of 4 functional groups, and 16 is 4 from each of 4 functional groups. And in this experiment, they looked at how much biomass was produced, as well as looking at beneficial insects, soil microbial activity, water balance equation, soil carbon, nitrogen and phosphorus. And there's been several hundred papers have published in international peer-reviewed scientific journals have come out of this experiment. One of the things that they did find was that as they increased diversity of the plant community, biomass increased. They also found, this is just an aerial shot of the Yena biodiversity experiment. These plots were 20 metres by 20 metres square. So where they're all the one colour, it's obviously they just have one kind of plant growing in them. A lot of them were flowers, and where they're kind of like that mottled appearance was where they were the multi-species plots. But one very interesting thing that came out of this experiment, and I'm just amazed that there hasn't been more notice taken of this in the scientific community, was that where they had 1, 2, 4, 8 or 16 different plants, and remember from 4 functional groups, and this was a multifactorial experiment with no nitrogen, or 100 kilos of nitrogen per hectare per year, or 200 kilograms of nitrogen per hectare per year, with every combination of every treatment. So you could have like a monoculture, say ryegrass, with no nitrogen, or ryegrass 200 kilos of nitrogen per hectare per year, right up to 16 plant species with no nitrogen or 200 kilos of nitrogen per hectare per year. Very, very thorough and well-planned experiment. What they found was that high diversity, in other words, 8 or 16 plant species growing together in one of those 20 by 20 metre plots, produced greater plant yield than high nitrogen. So if you just had one thing like ryegrass, or even 2 things like ryegrass and clover, two plant families, like ryegrass and clover, with 200 kilos of nitrogen per hectare per year could not produce what 8 or 16 plant species could produce with no nitrogen. And there has now been some research in Ireland that has shown similar results. Unfortunately, a lot of the research is still being carried out using nitrogen, and we really have to have that zero nitrogen treatment in there, because the whole idea is that we have to find ways of farming without using nitrogen. And the other thing was, now that we're talking about carbon, remember carbon and nitrogen are bound together in the same molecule. So we can't talk about one without talking about the other. But in this same experiment with 1, 2, 4, 8 or 16 plant species and the 4 functional groups again, they found that these high diversity plots, those are the ones with 8 or 16 species, actually accumulated 21.8% more carbon than the low diversity plots. In other words, 1, 2 or 4 plant species. So diversity, just plant diversity on its own, this is growing all these different kinds of plants together. This is with no nitrogen now. Just looking at the effects of diversity on its own is that high diversity plots are going to accumulate more nitrogen, and that means you're not are going to accumulate more carbon, which means you're not going to need nitrogen. It's all about finding ways of farming so that we do not need nitrogen. So the Yana biodiversity experiment showed conclusively that levels of both soil carbon and soil nitrogen were directly linked to plant diversity. And other studies have also shown that plant diversity is the greatest driver of the production and the stabilisation of microbial necromass. Now remember that microbial necromass is going to be our stable carbon that we can rely on being there for a really long time. So the species-rich plant communities have shown to be more productive and to exhibit increased long-term soil carbon storage. It's a shame that this diagram wasn't in colour, and it's not really very clear, and I also can't understand why they've put grasses here, but this is a recent article that sort of summarises a lot of the work that's been done on what they call plant species richness, which is basically plant diversity. Plant species richness, which is what's shown at the top of this diagram, leads to increased microbial biomass, leads to increased microbial necromass, leads to stable soil carbon. And this article is actually a review of quite a few articles that have been published over recent years on that subject. And an article that was published this month in Soil Biology and Biochemistry showed that 2/3 of microbial necromass, so now we're talking about stabilised soil carbon, originates from fungi. So our soil fungi are so incredibly important. Bacteria are important too, and bacteria and fungi are very closely related in terms of they work in teams. They help each other. Fungi can't function without bacteria, and bacteria can't function very well without fungi. But 2/3 of microbial necromass originates from fungi. In other words, 2/3 of stable soil carbon has been derived from soil fungi. So the fungal energy channel is also called the liquid carbon pathway. Basically, the fungal energy channel is what's shown in my first slide, which is we have the plant root over here on the left-hand side, we have soil particles over here on the right-hand side, and we have all these fungal hyphae inside the rhizosheath, which are just streaming energy from the plant out into the soil ecosystem. And in terms of nitrogen, one of the things that we do know about fungal hyphae now, where they're connected to plants, and these don't have to be symbiotic fungi, they can just be saprotrophic, opportunistic fungi that are beneficial. They're not pathogens, but they're connected to the plants, and they're moving energy out into the soil ecosystem to support bacteria and archaea. But they will have biofilms. There'll be biofilms on these fungal hyphae of nitrogen-fixing bacteria, because remember that soil microbes need nitrogen just as much as plants need nitrogen, and that where we have an ecosystem that looks like this in the soil, there will be a lot of nitrogen fixing taking place on the exterior surfaces of those fungal hyphae, because there is a little bit of root exudates actually leak out through fungal hyphae as well. So they kind of like have a leaky system in the same way as roots themselves have a leaky system with exudates coming out to feed soil microbes. The other thing about nitrogen fixers that we now have a lot more information on that we didn't have in the past is that we have endophytic diazotrophs, which I mentioned on Wednesday. Sorry about the big words, but they're basically nitrogen fixing bacteria that live inside plants and they get their energy directly from the plants. And they also have a low partial pressure of oxygen inside the plant, so it's very easy for them to use the nitrogenase enzyme to fix atmospheric nitrogen. So plants can get all the nitrogen that they need, provided they have these relationships with soil microbes, and adding inorganic nitrogen to that system causes a complete breakdown of everything that you're looking at there. It just causes total dysfunction in the soil. So again, this is another, just to give an indication of the fungal energy channel. We have the soil plant root coming down here, and all of this network that you see coming off this plant root are actually fungi, in this case mycorrhizal fungi, and they're transporting energy out into the soil ecosystem. They're bringing carbon and water. They're bringing water and nutrients back to plants, taking carbon out, bringing water and nutrients back, and a lot of trace elements and other things that are very important to plants. So we can't see these with the naked eye, but in a healthy soil, this is what the soil would look like. Would be totally jam-packed full of fungal mycelium. So this is why the use of fungicides is highly detrimental to soil function, because it is going to interrupt the very processes that we need to have healthy soil. So there are many reasons why soil microbial diversity is important. It's not just about carbon sequestration, which is the one that's shown on the bottom here, but it's very important for soil structure. And of course, you can't buy structure in a bag. And the more nitrogen that you apply and the more phosphorus that you apply, the more damaging effects you will have on soil structure, because root exudation will reduce. And so it's exudates that are going to feed the microbes that are going to produce the structure, or produce the glues and gums that make the structure, give you the aggregates, all of those sorts of things. And diversity is very important to that too. Microbial diversity, it's not just having an abundance of microbes, but we need a lot of different kinds of microbes in the system, and particularly we need representatives from the major groups like the bacteria, the fungi, the archaea. Very, very important for nutrient cycling, which I'm not going to talk about today because I'm just talking about carbon. But crop and pasture productivity is obviously what's going to determine the bottom line for farmers, as well as resistance to pests and diseases. So if soil microbial diversity is what is going to give us resistance to pests and diseases, that is going to be what will negate the need for fungicides. So again, the more fungicide we use, the more we will need fungicide. The more inorganic nitrogen we use, the more we will need inorganic nitrogen. So we have to figure out how to move away from using those products. Right at the beginning of today's session, Sean mentioned that the corporate world was benefiting from people being sick, and just the majority of illness that's in our communities today is the result of a very ineffective and dysfunctional food production system. It means we have food that's got low nutrient density, low vitality, low levels of really important, particularly secondary plant compounds that people need for health. And it's also loaded with chemical residues that have a further impact on our immune system. But, you know, corporate world is benefiting from this failure. And corporate world is also benefiting from a dysfunction in soils. So unhealthy soils mean that farmers have to apply more inputs to try and maintain production. But if we can turn that around so that we have healthy, resilient soils, those inputs will no longer be needed, and the corporates that are benefiting from dysfunctional soils will just hopefully go find something else to do. So it's going to be all about supporting microbes rather than using high analysis fertilisers. And the two main ways to increase microbial diversity are plant diversity, which I've already mentioned, and the use of biostimulants, which Jerry Gillespie is going to talk about as part of his talk, which is going to be following mine this afternoon. So Irish on-farm experience has already shown that diverse forages can not only replace but can outperform low diversity swards plus fertiliser. So, you know, the evidence is already there. It just amazes me that it's taking so long for these changes to take place. The tide has turned and it will not be possible to hold it back. So I thought my presentation was probably a little bit heavy today, so I've just thrown these in for a little bit of lightheartedness at the end. We may think, if we're on the, what's it called in the, on Mount Stupid, we may think that we can turn back the tide, but unfortunately we can't. We cannot. Nature, that's last, and nature will win. So high analysis fertilisers are merely a substitute for plant diversity. They are absolutely not needed in agriculture. And in situations where people feel that they can't immediately go into a diverse situation, for example, grain crops or your tillage farmers who feel that diversity is something that they need to know a little bit more about before that they can start producing multi-species cash crops, for example, the use of biostimulants in place of high analysis fertiliser is an option to take you part of the way there. And in fact, if you can combine biodiversity and a biostimulant, then it's can produce absolutely amazing results. A biostimulant is not a biological fertiliser. It is something that will stimulate biology, which is why it is called a biostimulant. And then that biology, which is basically our soil microbes, will then do the work of accessing nutrients, protecting plants and building soil. So this is our little durum wheat seedling. The seed is just here. It's only got 2 leaves. You can't see the leaves in this photograph. It's produced this amazing seminal root system going down, 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 down. Amazing riser sheaths on the crown roots, and all that has had on the seed is a biostimulant, which was a combination of vermi-liquid, in other words, from a worm farm, and compost extract. So that's the end of my formal presentation today. I have left some time for Q&A, and I know that this presentation is going to be followed by Jerry Gillespie, who will speak about biostimulants and other things about recycling in much greater detail than I have. So
much.
That's it from me.
Thank you so much. That's great. Unsurprisingly, we have an awful lot of questions for you, Christine. So we're going to dive straight in and get them answered, if that's okay with you. So we start with Coleman Deely. Are there any damaging side effects to plants and/or gut health, he asks, from protected ureas above other artificial nitrogen products?
I can't answer that question, Ella. Sorry.
That's okay. We'll move on.
Yes, I don't know what the effect would be on human health.
Okay.
Well, was that a question on what effect would they have on human health?
Well, actually, yeah. Well, to plants as well. So, you know, protected urea, the sort of impact of protected urea on plants.
Yeah, I couldn't quite understand why it would have an effect on gut health. That didn't. I mean, we're not going to be eating it, are we? I mean, the reason for protecting urea is to reduce its mobility and to have it release more slowly. But my question would be, why are we using it at all, really? I mean, why not use organic nitrogen? If you need to use a nitrogen source, why not use something like fish hydrolysate, which is absolutely like totally safe? Like, can we please get away from using inorganic nitrogen? There are organic sources of nitrogen that you could use that are totally safe, that are not going to hurt anybody. You could probably eat it yourself. But no, I'm not a fan of protected urea. So
you advise farmers not to use it?
I would advise farmers not to use any form of inorganic nitrogen at all.
Okay. David Noel Murphy asks, does Christine see any role for humates, or is it only beneficial with inorganic nitrogen?
Again, no, I don't see any particular role for humates. I think that the reason that people use humates, this is purely my opinion, is because they consider that it's made from, like it has humic, they do have humic substances in them. Usually they're made, in Australia, they're made from things from like liardonite and brown coal and those sorts of things. They contain humic substances, and people make this leap of faith from humic substances to humus. Again, that's something I cannot see the benefit of humates personally.
Okay.
And I have never seen really any research that would show that there's a benefit to those.
Norman Dunn asks, hi Christine, what is the potential for methanotropic microbes in a soil, and are there any specific things we can do to encourage them?
That's a great question. I love that question. Thank you, Norman. A lot of the work that's been done on methane in Ireland, and I was going to put a slide in, and then I thought, no, it's too far off the track to talk about methane. Animals have had special, like, apparatus, like fixed, cows have had things put around their faces and everything to measure the amount of methane that they're breathing out. Or they've been put into metabolism cages to measure how much methane they're producing, which is a totally unnatural situation, because when they're grazing out in the paddock, there are microbes in the soil, which Norman has mentioned, that are called methanotrophs, and their sole energy source is methane. They can only use methane for energy. They can't use anything else. So any methane that's in the atmosphere, they will grab that and metabolize that and use it for energy. And as they are using methane from the atmosphere, they're drawing it down into the soil. You get like a concentration gradient, and methane comes down to fill that void. And they can, if the soil has sufficient methanotrophs in it, there is no net emission of methane to the atmosphere from grazing animals. That is not going to happen in a metabolism cage, and that is not going to happen when you have animals with some kind of apparatus around their faces that's just collecting the methane that they're breathing out. That's totally illogical, and I think actually quite deceitful to talk about how much methane an animal produces when it's in a totally unnatural situation, rather than looking to see like we have methane sinks as well as methane sources. Obviously ruminants are a methane source, but the methane sink is the soil.
So how do you increase
again,
them?
If you actually use nitrogen fertiliser, you wipe out methanotrophs in the soil, that they're completely intolerant to nitrogen fertiliser. So again, you would have to have a system where you weren't using nitrogen fertiliser in order to have good levels of methanotrophs in the soil.
Okay, so that's very clear. It's more about what not to do rather than to do. So Ian Gould asks, what do dairy farmers do with their slurry if they don't need the nitrogen from it?
Jerry Gillespie is going to talk about some very beneficial things that dairy farmers can do with their slurry. Following on from my presentation, he's specifically going to speak about that.
Okay, Martin asks, does Dr. Jones have any comment on using worm extract and milk to control diseases instead of fungicides?
Yes, milk is very beneficial provided you use it early enough, and I'm presuming that you're talking about using it as a foliar. So if milk is applied to the leaves of something like, say cucumbers or zucchinis or whatever that tend to, late in the season, we'll often get fungal spores will accumulate because they've got quite hairy leaves, and they'll get caught in the wind and they'll start to multiply on the leaves of those plants. If you catch them early enough, milk will definitely have an antifungal effect without being detrimental to soil microbes or microbes on the plant.
Okay.
And what was the other thing? And worm extract, yes, definitely. Worm extract or vermiliquid is beneficial, is extremely beneficial, in fact, to put on seeds. That's what we need to be putting on seeds rather than putting fungicides on seed. Is if we coat seeds with vermiliquid, they produce much stronger seedlings that grow faster, that have greater resilience against pests and diseases, and particularly against fungi. That's what we should be putting on seeds instead of putting fungicide on seeds.
Okay, Robert asks, does Christine use SAP analysis to monitor crops, soils, etc.? What is her view on the use of SAP analysis?
I don't, and I I would suggest that it's probably beneficial if you're in a very high value horticultural industry situation where you're producing very high volume, rapid turnover products, like if growing vegetables in an environment like in one of those, I don't know what you call them in Ireland, I'd call it like a glasshouse kind of a situation. And you want to know, what micronutrient levels are in plants, and you need to know like very quickly, then I would use SAP analysis in a situation like that. I think for farmers, if you're a dairy farmer or a crop producer, tissue test is adequate, is perfectly adequate. And in fact, I would recommend that people would always do tissue tests, and that should be like, I can't imagine how you would even not be using tissue tests. I wouldn't bother with soil tests, I wouldn't bother with SAP tests, but I would be using leaf tests or tissue tests, whatever you call them in Ireland.
Okay, thank you. Martin asks, would an EC meter be of benefit to show how well your microbes are performing?
I don't think so. I would just use a refractometer. I think the only tools that a farmer needs is a spade to dig a hole, to have a look at the roots, to see whether the plants have riser sheaths, and a refractometer to measure Brix levels to see whether the plant is actually photosynthesizing fast enough to be able to support a diverse microbial community. And then if I was going to send anything to the lab, I would send some plant leaves and have a leaf test or a tissue test done on those leaves. They would be the three things that I would consider to be important for monitoring how things were going on my farm.
Now, Sean asks, how damaging are seed handling systems to the microbes on seeds? Can we also ask whether seed treatments to the seed crop affects the natural biology of the seed that's produced?
Yes, seed handling, I'm not quite sure whether you're talking about like bulk handling. I think probably seeds are fairly resilient in terms of how they're moved around, but certainly the way they're stored, whether they're stored with insecticides, which is quite common, and then we have seed dressings that contain insecticides and fungicides, which are highly detrimental to the microbes that are inside the seed. So as the seed germinates, it's going to have a core microbiome of endophytic microbes that move out into the soil from the seed. And if those endophytic microbes encounter toxins like insecticides and fungicides in the immediate vicinity of the seed, they are not going to be able to communicate adequately with resident soil microbes, which are then needed to move back into the germinating plant and protect it from things like fungal pathogens and insects. So we basically prevent the plant from defending itself by putting poisons on the seed. So how we actually handle the seed in terms of what chemicals we put on it is very important. I don't think how we handle in terms of how we move it about is important, but how we store it. I mean, there is no reason why seed can't be stored in situations where we can make the storage airtight, and then the space can be filled with nitrogen, which is much cheaper. You can, at one time people used carbon dioxide for seed storage, but actually nitrogen is much more efficient, it's much cheaper, and there's no way that things like weevils and other pests can grow in an environment that doesn't have oxygen in it. So we've excluded oxygen from our storage facilities. We don't need to use any poisons in the storage of seed.
Okay, and there's more questions. We'll come to them in a minute. And anyone else who's got questions, please put them in now. Sean. Hi,
Christine. Yeah, it struck me, Christine, when you're talking about your 4 families and the more diversity in the sward. Does it really matter then, if you have the 4 families, what plant you use? I'm struck by the fact that Sean Condon yesterday has reached the conclusion that his old permanent mixed almost natural grassland is quite productive and effective, and he doesn't see the need for things. And then if you look at, we'll say, the likes of James or Maya, they're on this continual multi-species. Is it more so because they're coming from almost a monoculture of ryegrass that depended on high nitrogen, and they have to move off that system, and multi-species seeding is the easiest way for them to do that?
Well, that's sort of a two-pronged question, Sean. The thing is that if you have come off a monoculture situation like a high input, very low in microbial diversity, very low in microbial abundance, the quickest way to restore that would be to have as many different kinds of, or as many different functional groups of plants as you could put. So at least 4 functional groups, which is why I say 4 plant families, and then as many species within that as you like. But we have seen quite amazing effects with just 4 plant families, like just 1 species from each family or 4 plant families seems to be incredibly productive. Sometimes people think they have to have lots of species, and that's why, rather than lots of families, I mean. So people might have 6 different kinds of grasses and then 6 different kinds of legumes, and they think they're going really well in terms of diversity, but they've only got 2 functional groups there. So I really have to emphasise that it's the diversity in functional groups, because each functional group is going to have its own suite of microbes associated with it, and it's microbial diversity. At the end of the day, it's microbial diversity that drives the system. And the quickest way to get that, if you're in a dysfunctional system, is to have plant diversity, because each kind of plants, each family of plants supports a distinctive microbiome. Now, in the situation that you talked about, like a grassland, which is called a grassland, but in fact, if you go and have a look into it, you'll find that there are a lot of things besides grasses in there. It won't just have grasses in it. I'm sure it hasn't just got grasses in it. There'll be, I'm sure he's not spraying out everything that's not a grass, for example. Like whatever wants to live in there is living in there. And I think if you go and have a really good look at that, that ground cover that is there, you'll probably find there's 10 or 12 plant families represented there. And sometimes some of the plants are only small. We hardly even notice that they're there. But what we've discovered, or what researchers, like not me, but in the ecological literature, what we see is that it's sometimes the very tiny plants, the things that don't contribute very much to biomass, the things that are just down on the soil surface, or they might be things that kind of, like stoloniferous or rhizomatous, and they move around in between other plants, don't contribute very much to the bulk of material that's there, but they do contribute significantly to microbial diversity, and they do make a significant difference to productivity. And I have seen that like firsthand. If we go up into Northern Australia, some parts of Northern Australia are tropical, and some parts are extremely hot and dry, and definitely not tropical. So we have what we call the dry tropics. So it's hot, but it's not wet. And it's, when you look at how productive some areas of the dry tropics are, it's because of all these amazing little tiny plants that are in amongst those. There'll be grasses there. And if farmers are managing those grasses in such a way as to maintain good ground cover, like not overgrazing them, not stocking and destocking in droughts, and doing all the right kinds of things to maintain what they consider to be a grassland, when you actually look into that grassland, there are basically hundreds of plant species in there. And there is a lot of diversity, but a lot of them are little tiny things that you really wouldn't take much notice of unless you got down on your hands and knees and really started to look. So I wouldn't be surprised that in some of these long-term grasslands that people think that all they have is grass. It'd be interesting to send a botanist there and find out what else they have there.
Okay, thanks.
Thank you. Another question, Christine. Ian asks, if using vermi-liquid or compost extract, should you use it undiluted, or can you water it down to make it go further?
You would definitely water it down. Well, I guess it depends what you're going to use it on. If you were putting vermi-liquid onto seed, for example, you'd put something like about 5 litres on a tonne of seed. So you're not watering it down to put it onto the seed, but you're not putting very much on the seed. 5 litres on a tonne of seed is not very much vermi-liquid. That's about the rate, pretty much the standard rate that's used. So if you have, 200 litres of vermi-liquid or something, it's going to cover a lot of seed.
Okay, Klaus.
Hi, Christine. Thank you so much again for sharing all your knowledge. You're such an inspiration. The only problem I have, everybody should listen to you. All farmers should be aware of what you're saying. I think it's so important. You've inspired me, was it a couple of years ago at the BioFarm, and I've done a 16 vegetable salad mix, which was great fun and highly productive and all, you know, mixing all the different vegetables, annual herbs together. The only problem is it's not, I couldn't work that in a commercial basis, you know, but as a hobby gardener, it's such fun. I have a couple of questions for you, Christine. One is the use of biochar. I used it in the last few years, and I feel it's really working wonders in heavy soils and to loosen it up. I haven't done experiments. I've read a few bits about it, but what's your experience about biochar?
Again, it depends on how you manage the biochar, because as you're aware, if you look at it under a microscope, it's got like a honeycomb type appearance. It's like sort of like a sponge. And if we put biochar into our soils here in Australia without preloading it, by preloading it, I mean if you put it into a compost heap so that all the little holes in the biochar are filled with microbes from the compost heap, or if you feed it to a ruminant animal and then you collect the manure, so it's actually been through a living system, that's what we call loaded. When it's loaded, it's very beneficial for the soil. But if we just make biochar by pyrolysing biomass and then add that to soil in our environment here, where our soils are very nutrient poor, and because the biochar, as I said, has a structure like honeycomb, it basically sucks all the nutrients out of the soil and deprives plants of the nutrients. So the official trials that have been done by the Department of Agriculture, where they've looked at biochar, the government actually spent $2 million doing biochar trials, found that yield reductions range from about 5% to 35% where biochar was added to the soil. And then it was discovered that that was because it sucks up the nutrients out of the soil and actually deprives plants of the nutrients. That's not going to happen in a situation where you're using compost or you're adding manures or you've got very biologically active soils. But in our nutrient poor soils and our very light soils, a lot of them are very sandy. They're not heavy clay soils like what you're probably talking about. It actually was detrimental. But then people discovered that if they mixed the biochar into compost, or they put biochar through, just fed it to animals and then used that manure, that was fine, because then it's loaded with nutrients, and it's a great repository for nutrients in the soil.
We have a question here from Rachel. Would Christine be able to provide a bibliography that Knots could share relating to the research she's presenting? Where are the key places to start studying?
If somebody would like to send me a list of things that they would like references on, that would certainly be absolutely doable.
So to save you from getting multiple emails, perhaps you could do that. You could send it to Sean, and then we can you put it up on the site, Sean?
Yeah, we can. Yeah.
That'll save you from getting the same request multiple times, Christine. So Sean will put it on the site if you could send it to him. And finally, Paul asks, Sean, when is Christine coming back to Ireland for a course? And I presume that's a question for Christine, not Sean.
Christine and me discussed this on Monday, I think, Christine, wasn't it? So as soon as COVID settles and everything is grand, Christine is coming back. We've talked already about this.
Is that a fact, Christine?
At the moment, Australians are not allowed to travel. And if for some reason we found a way to get overseas, we're not allowed to come back. So our federal government has restricted our travel for the last 2 years. We call it Prison Island. We're not allowed to leave and we're not allowed to come back. But when the airways are open again, yes, it definitely is a fact that I would love to come back to Ireland.
Well, Christine, thank you very much for the last hour, and thank you for answering all the questions as well. It's been obviously very stimulating for the audience, and we look forward to welcoming you here in Ireland at some stage in the future. But we are going to be back with Jerry Gillespie in about 5 minutes. So stay with us, and Christine, take care and good night over there in Australia.
Thank you. Thanks, Ella. Thanks, Sean.
this.